Hey everybody, back with another review. This one's for DNA and genetics. I'm just going to give you a quick 5 topics. The topics are 1, DNA and RNA, 2, what are genes, 3, the central dogma, 4, mutations, and 5, Punnett squares and inheritance patterns. I'm just going to warn you up front, I'm just going to kind of scratch the surface on all of this. If there's anything that comes up, like any vocabulary or topics that you don't quite understand, I'm going to highly encourage you to go look them up. I'm only doing the very, very basic stuff here. So first off, DNA and RNA. DNA, it's a double helix shaped molecule. It looks like a twisted ladder. It's made up of individual molecules called nucleotides. Those nucleotides each have 3 parts, a phosphate group, a deoxyribose sugar, and a nitrogenous base, either adenine, thymine, guanine, or cytosine. You'll usually find DNA inside of the nucleus of a eukaryote. For things that don't have a membrane-bound nucleus like prokaryotes, we call the place where you find the DNA the nucleoid space. And the role of DNA in cells is to store genetic information. RNA, typically single-stranded. It also is made up of nucleotides, but with a couple differences. Specifically, the deoxyribose of DNA is replaced with ribose sugar, and nitrogenous bases, we're going to flip out thymine for uracil. It comes in 3 types: mRNA or messenger RNA, tRNA or transfer RNA, and rRNA, ribosomal RNA. They all serve different roles. And RNA is used throughout the cell to carry and interpret genetic information. So what are genes? Basically, genes are segments of the DNA that carry the instructions for a particular cell product or function. So an easy way to think of it is like recipes in a recipe book. Each recipe does something different. And so if you want to make a particular type of cookies, you're going to look for that particular recipe. Now, genes are passed down from parents to offspring, and in that process, you may have mutations. I'm going to talk about that more later. It's a really important part of understanding evolution. Genes are found on chromosomes, which are larger segments of DNA found within the nucleus or in cells. And if you put all the genes together, all the chromosomes together, you get what's known as the genome of that organism. The central dogma. It's a way of understanding how DNA is read and interpreted in cells to make products. That product is usually a protein. So the steps, first we have transcription, where the information in DNA is copied into a temporary mRNA molecule, in the case of eukaryotes, so it can leave the nucleus. And then translation, that mRNA is read and interpreted in the ribosome, amino acids are put in the correct order, and you assemble the final protein. Replication is often mentioned as part of the central dogma as well. Replication is the copying of DNA exactly. Cells do this right before they divide. Now, sometimes that copying doesn't go perfectly and we end up with some errors. These are called mutations. What this does is it can alter the product or when the gene is read. So think about it. If you had a recipe for cookies and you subbed out sugar for salt, you're going to end up with something very different. Now, if they occur in the DNA of a gamete, which means a sex cell like an egg or a sperm, that mutation is going to be passed to offspring and it's going to be present in every cell. One thing you have to understand, though, is that mutation is not always a negative thing. In fact, mutation is what drives evolution. So a mutation that makes you run a little faster or have an easier time reproducing is going to be passed on more readily. Some types of mutations, we have deletions. Deletions is where there are nucleotides missing. Substitutions, nucleotides are swapped out. So instead of having an A, maybe you now have a C in that location, which can affect the recipe. Insertions, extra nucleotides, and duplications, sections of nucleotides are repeated over and over again. There are larger mutations known as chromosomal mutations, and they affect entire sets of genes. So instead of just making a tiny alteration in one recipe for cookies, we're affecting the entire desserts chapter of the big cookbook of the genome. Some examples of chromosomal mutations you may hear of are translocations, where parts of chromosomes are swapped, and inversions, where a piece of a chromosome is flipped. Finally, our last topic is Punnett squares and inheritance patterns. This is actually a really big topic, but we're going to do a little bit of it. So if you remember, a Punnett square is a diagram that helps to show the probability of the genes an offspring will receive based on parental genes. So what are the genes of the parents? What will the genes of the kids look like? So a couple important vocabulary terms. Genotype refers to the combination of genes seen in an organism. So in this case, if we're talking about Mendel's famous peas, flower color had two options: purple, which was dominant, to white. In this case, if we have one gene for purple and one gene for white, this organism will look a certain way. But for us to indicate that combination of genes, we're going to use letters. Big P for the purple gene, little p for the white gene. Phenotype is the result of that genetic combination. So genotype controls phenotype. In this case, because there's one purple gene and purple is dominant, all of the flowers of that plant are going to be purple. Homozygous, that means that the two genes in an offspring in a zygote match. So basically, if you have an organism, a pea plant that has big P, two purple genes, that's homozygous purple, or little p, two white genes, that's homozygous white. Heterozygous, that means the two genes do not match. So here, a big P and a little p together would be deemed heterozygous because we have a purple gene and a white gene. If you remember how Punnett square works, we put the genotypes of the parents. In this case, both parents are heterozygous. These are birds. They have blue feathers. They carry one blue gene and one yellow gene each. We're not seeing any yellow in the parents because the yellow gene is being overwhelmed by the more dominant blue gene. However, when you start to put their kids together, you get some interesting combinations. Big B, only blue gene's going to produce blue. Big B, little b, still we have a blue dominant gene present, so we're only going to see blue feathers. And then that little b, because both genes are recessive, even though they weren't seen in the parent because there was a blue gene present, now we only have the recessive gene. So these birds are going to have yellow feathers. They're going to show the recessive phenotype. So phenotypically, if these two birds got together and had babies, we would expect 75% of their offspring to be blue, 25% yellow. Is it possible that their offspring could be 100% yellow? Absolutely. Every offspring has the chance of being any square in the Punnett square. But probability states that about 3 out of every 4 will probably be blue and 1 out of every 4 will probably be yellow. Okay. Genotypically, we expect 25% of their offspring to be this double dominant homozygous true breeding blue. We're expecting 50% of their offspring to carry the yellow gene but not express it, and then 25% of their offspring to have the recessive phenotype because they're homozygous recessive. Okay. So some inheritance patterns you'll see. The one we just described with that Punnett square is known as complete dominance, where one gene is more dominant than the other. As long as that blue gene was around, you won't see yellow. So heterozygotes take the dominant form and you can't tell them apart phenotypically. Incomplete dominance is where one gene is not dominant to the other, they're equal. And so when both genes are present in a heterozygote, you actually get something in between phenotypically. So if blue was incompletely dominant to yellow, chances are we'd have a bird with green feathers. Codominance is similar, except this time they're both dominant, but if you put them together, you actually see both phenotypes simultaneously. So instead of blue plus yellow equals green, blue plus yellow equals yellow and blue. Sex-linked inheritance is basically a gene is only found on a sex chromosome. Depends on the organism. Fruit flies and birds and humans have slightly different versions of all these things. But for example, in humans, if you have a gene only found on the X chromosome, someone who has 2 X chromosomes is going to have a different inheritance pattern than someone who has 1 X chromosome. Polygenic traits, these are traits that basically you see a phenotypic spectrum. There's no this, or even just 3 options. You have an entire rainbow of variety of options. So things like human hair color, skin color, eye color, where, for example, in eyes, they have everything from the darkest brown eyes all the way up to the lightest, palest gray eyes with every sort of green and hazel and brown and blue in between. That's because polygenic traits are controlled by more than one gene. All right, I hope that helps a little bit. Again, I know it was very brief and you may want to go look up some of these topics and learn more about them, but hopefully that jogged your memory.